Good afternoon, everyone. My name is Franklin. I'm going to be talking about the work that some of my group is doing on defense systems and their activity in E. coli. But before we get started, I would like to say, please do not miss out the work that my group is doing. Some of them is their first conference, and they'll be very happy if you could just spend some minutes talking to them on their posters. And you can see there's quite a lot of diversity of work that we are currently doing. But changing gears to the topic, I'll be talking to you about bacterial defense systems. As probably many of you know, bacteria will find many different ways to actually cope with the mobile genetic elements that are constantly trying to get inside of them, plasmids, or as we probably are very much aware here today, phages. There are many different defense mechanisms that are known. This is probably the schematic to me that probably highlights best the mechanisms that are quite diverse from which these bacteria fight. You can see on the top CRISPR-Cas systems, but of course, restriction modifications is still the system that probably bacteria use the most to be able to cope with a lot of the phage pressure that you see. However, nowadays, there's 110 defense systems. Of these systems, there's 201 subtypes. And all of these systems, they occur in what are called defense islands. These defense islands are regions that are non-randomly located into the genome where different defense mechanisms are being found. And what these systems are they're doing in this composition, in this diversity that we find in these islands is not yet very well understood. So my lab tries to understand how these combination of defense systems are being selected against or for anti-phage protection. And for us to do so, we used E. coli as it's one of the model organisms that we have in the lab. And for that, we just pull down all the bacteria that were present in public databases, and we pull down 26,000 genomes, and we use two of the tools that are the reference for look for anti-phage defense systems. I have a preference from one of them. I don't know if you know why, but anyway, I have a preference for the tool. However, Padlock and Defense Finder are the two tools that you can use to find new defense systems. And like many of my colleagues that work on this field, we're able to plot the diversity of these defense mechanisms along the chromosome, but also along mobile genetic elements. As you can see, this is just probably seen by many of you before, but what is quite striking is that you can see that some of these defense systems occur in mobile genetic elements, and this is the case of the BSCA, actually found here in a lab in the UK from Jay Hinton, where actually found a new system that is a new type of superinfection exclusion. However, what we were able to see is that some other defense mechanisms are occurring in plasmids. And today I just had a very nice talk about Gabaya, which is one of the systems that we actually can find very frequently occurring in plasmids. This is an abortive infection mechanism that is actually being targeted and across moving along plasmids. And you can see that in E. coli, but also on Salmonella typhimurium, if you're particularly interested in that pathogen. What we really wanted to take a step further was not only to quantify signal, but trying to understand what it means. And what it means is, what does it mean that we have an average 5 or 6 defense systems in a given strain? So for that, we wanted to understand how does this co-occurrence happens more frequently than random chance. So in other words, we wanted to look into positive co-occurrence and how that positive co-occurrence have an effect into a certain phage attack. And this is what you see here in this matrix. In orange, you see these positive co-occurrence when comparing to random chance, having a certain defense system, the cooperation present in one strain. So for that, we designed a very elegant system where we had a 2 plasmid system that allows us to have a control plasmid and 1 defense system, or 2 defense systems at the same time. And this is what we're going to try to show you, or I'm going to try to show you, is that you have 1 defense system on top with a YFP protein control being expressed at the same time. In the middle, you have the other system with the control. And in the bottom, you have 2 defense systems where you can see the defense. And what I hope I can call your attention to you today here, and hopefully online as well. If you look at T1 as a given example, you can see that T1 Gabaya doesn't challenge that phage very well. So the system is not providing strong protection. TMN is not providing strong protection, but actually the cooperation, the co-occurrence of those 2 defense systems is giving a synergistic effect of protection, and is the combination of the 2 defense systems in the strain that is providing an anti-phage activity, as you can see by the here. And you can see by that, by all the orange ones, where you can see the synergistic interaction. However, it's important to realize that not all the interactions that we see are synergistics. And this is actually where you can see that by on the arrows. You can see T7 and Phi113, where actually that effect comes from the single system and not from the combination of those systems. But of course, when we actually were able to see this, we were quite happy to see these results. So we wanted to understand what has happened when actually you have something that is negatively occurring. When does it happen when you actually have a system that occurs less frequently than expected? So we wanted to have an assay where we could look if there's mechanistic incompatibility between defense systems, and that's the reason that we don't find them frequently occurring on the same genome. So we put some of these pairs, and for our surprise, they don't provide an antagonistic interaction, suggesting that there's no mechanistic incompatibility between them, but there's probably something else happening that is favoring these defense systems. And we found this in other pairs that we study by looking at the same less frequent occurring pairs. Of course, every single thing matters when you're looking at very big data sets in the certain type of species, but will these data look the same if we're looking at that taxa? So what we did was picking up other types of taxa and looking if the co-occurrences that we have just found were showing the same pattern. However, we could see that some of them, although do co-occur, sometimes in a negative correlation, it becomes a positive correlation for the other taxa, demonstrated actually that these negative co-occurrences are probably deriving from environmental factors. So we have conditions from which species are being selected for containing certain types of defense systems over others. When we start looking at defense systems, we want to understand mechanistically how they work. So for that, we look into phage escape mutants. We want to understand what are the sensing, the trigger mechanisms that the bacteria are sensing to have a certain phage responding to it. But we also want to understand how does the phage overcome that defense mechanism. And I bring here one of the mutants for that. It's a T3 mutant into our combination of the 133 and Zoria2. And what we're able to demonstrate is actually this combination is detecting a tail protein during the infection process. What I really want to show and guide you through is our ecological implications of our findings. So if you can see here, it's a plot of fold change to the wild type. So if our mutant phage is performing better than the wild type, you'll see the result higher than 1. But if it's the other way around, you see a result lower than 1. And what you can see here on the plot is that you can see that our mutant phage is performing better than the wild type in the combination, not in a single system, but in the combination. But what is more importantly to look is actually when we start looking at other combinations that we have seen before. So we're able to demonstrate that some other combinations are able to then target this mutant and drive that mutant to extinction. So if the phage is evolving to overcome a certain defense combination, then the other defense system combination is targeting that phage mutant, driving that predation to 0. However, and more interestingly to me, is that if you look at the negative pairs that we studied before, you can see that our mutant phage is able to overcome that negative pairs as well. This doesn't mean that the phage combinations are not providing synergistic effect and they're not providing protection against the phage, but this is able to demonstrate that it's actually the environmental factors that have been showing before is driving the selection of defense systems. Because we can generate mutants so easily, these mutants can possibly be generated in nature, so these defense combinations will not be selected throughout evolution. And those are just beautiful arrows on the bottom. On the end of the plot. However, phage host interactions are not ending without this talking about anti-defense proteins. One of the most favored anti-defense proteins are actually DNA mimicking proteins. T7 has an OCR protein, probably if many of you are known by now. And this actually found to stop defense systems like restriction modification or BRACs. So when we did this assay, we wanted to find, well, probably just going to find a bunch of different systems that are able to not protect against a certain phage infection because you'll have a DNA mimicking protein. However, that's not what we saw. What we see is that the DNA mimicking proteins are able to stimulate the bacteria to target the phage better. So we found that the OCR stimulates defense systems like Gabaya and Zoria. But more importantly, we found that in other cases, the systems are creating noise to each other. And this is the case of Druanthia and Zoria. We believe that when Druanthia is present together with Zoria, although it's a combination that we currently see, if the OCR is present, somehow it creates a noise in the sensing mechanism of these two defense systems that actually, it creates such a noise inside the bacteria that the bacteria, instead of being able to target the phage is able to overcome the system. So I hope that I've convinced you that negative and positive interactions provide synergistic effect to have antiphage activity, that these are not conserved along all the taxa, and that some of these selective pressure are not because of mechanistic incompatibilities, but because of environmental conditions like the panimmune model seems to suggest. I hope that I was able to also demonstrate to you that anti-defense proteins not always help phages to overcome defense mechanism, but in sometimes actually the cue that the bacteria uses to target the phage. And more importantly, that phages are exploiting single mutations. They're exploiting this single defense mechanism sensing capacity that the defense systems share to overcome many of these defense systems. And I hope with that highlight the complex coevolutionary dynamics of phage defense and anti-defense. And with that, I would like, of course, to thank my team, more importantly, EU, which is the lead on this paper, and saying that we are hiring. And if you're interested in law enforcement and developing a new forensic methods, please do reach out. And thank you very much for your attention.
Great talk, Gilde from New York again. I was thinking, like, do you think then there's some sort of like diminishing returns in terms of like number of phage defense system you can then have, because like over the time, you know, you become more sensitive, the likelihood of becoming sensitive to one phage increases. So you end up some sort of optimum and not like endlessly acquiring like 50 different defense systems.
Yeah, I think this is a very interesting question. I must say that recently I saw a poster from Joe Bondi's lab, and he said that he had more than 20 different defense systems on one single strain. And when I saw the poster, I was just wondering, how is that possible? But I think you answered somewhat that question as well. I don't think that all defense systems will have the same cues, and they probably will not be activated all at the same time. I think in the cases that we have been studying, we've been lucky to look at these synergistic interactions and see some probably mechanistic compatibilities. But I do think that some systems will be regulated by WYL as a given example. I don't know if that regulator makes sense to you, but is Tim Blauer showing that together with Plex, but quorum sensing and many others are probably playing a role into this response. So I think if the conditions are right, and if you're probably if the species have the ability to probably sense many different things, maybe you have many of them, but we are still very puzzled by some species that don't seem to have any of the known defense ones. But I just know at least of the 3 other labs that are just saying that they just found 20 new defense systems. And I wonder if nowadays anything is not a defense system. So I don't think it's probably around in the middle, if that makes sense.
Do you think, Franklin, you'll have situations where phages will work together to cooperate over, you know, to get our own defense system? So one will provide a part of a response and another one in a synergistic way.
In terms of the phage,
yeah.
I do think that that's very much possible as well. I do think that Eduardo Rocha and David Picard, they suggest for the first time the monogenetic element warfare. And I would say that probably phages will have, probably after domestication of the prophage as well, probably you don't know where it comes from, but I think probably we don't talk too much about phage interactions, and I do think that's probably playing some role in terms of the dynamics that we see.
Okay. Oh, question there, Pablo.
Oh, sorry. We can rocks, paper, scissors for it. This was a really interesting talk. Thank you for it. I'm Emma from Folium Science. I was wondering if you noticed when you were doing your analysis if certain strains that had certain pairings of these defense mechanisms also had prophages embedded in their genomes. If you noticed similar frequencies of prophages or none at all, or if you didn't look at all.
Yeah. So on the preprint that we released, that we have a quantification of all the prophage within these, and we know which systems are present there. We also know the ones that are present in plasmids as well, and some of them are rich. So Gabai is one of these cases where we look into the plasmid region, but some of them, as others suggested before, like BBSTA, the superinfection exclusion, is probably quite enriched in our data set, and present on what we believe to be active phages, but active prophages, but we don't know because we don't test them experimentally.
Looking forward to reading it.
Thank you for the talk. I'm just curious, I think you gave the answer on your talk. You had a sentence about the combination of all the different defense mechanisms, but at the end of the day, the bacteria has a lot of advantages by being infected by these phages, right? So there has to be a check and balance of what you're regulating. It makes me think of Klebsiella, the fact that if you lose the capsule, the bacteria regain the capsule by acquiring the genes that allow for that complement for the synthesis of the capsule, right? So the bacteria does want to regain the capsule for survival purposes. So there has to be a check and balance between all these defense mechanisms, right? So wouldn't that be why you have so many? And I think the sentence you said something about environmental factors playing a role. I don't know what your thoughts are on that.
Well, I think it's a great question. I must say that I would like to say that I only know that I can only speculate, right? If you think about like how CRISPR works, which is an adaptive immune system, you can see that if you have a self-targeting event going on, it kind of cleaves its own genome, so it will lead to cell death. And leading to cell death, then you know that there's a selective pressure going on. So in some of the other bacterial defense systems, we see that there's a lot of this question that many of them are doing abortive infection. So you see that a lot of the population is driving themselves to extinction and surviving. So then the question comes in, are we simply just selecting for the cells that are surviving that have never been infected because the population as a whole has been defended? Or you do have some kind of mechanistic incompatibility, as I said before, as CRISPR, because you're targeting some island is moving along and then you're selecting for that. I would say probably both are happening. And I think it depends on what kind of environment you're selecting for, I would say. But very happy to chat more about it if that.
I think in the interest of time, thank you very much, Franklin. That was an absolutely excellent talk.